Our next speaker is Adam Mosier, and Dr. Mosier is an associate professor in the Matilda R. Wilson Endowed Chair of Large Animal Clinical Sciences at Michigan State University. He received his BS in Veterinary and Animal Sciences from the University of Massachusetts in 2000, and he went on to obtain his MS degree in swine nutrition and a PhD in gastrophysiology, gastrointestinal physiology in 2006, and a DVM in 2008 from North Carolina State University. And after all those years in school, then you decided to do something with all that knowledge. And he joined the faculty at NC State in the College of Vet Medicine and was in that position for 7 years. And then just a year ago, joined the faculty at Michigan State University, where he's a named Chancellor's Faculty Scholar. Dr. Mosier's research is focused primarily on pathophysiology and stress-induced gastrointestinal orders in animals and people, and today he will be presenting on the topic of environmental influences on gastrointestinal development, function, and disease resistance. Please help me welcome Adam.
Okay. All right. Thank you, Steve, for the introduction. And as the title indicates, this morning I'm going to be talking about stress and GI function in the pig. And it's going to be mostly focused on the early life stressors that pigs undergo, and really what are the long-term impacts that they have on the gut. So we know that diseases, any diseases, particularly GI diseases, involve complex interactions between the host, pathogens in the environment. And a lot of the research and emphasis has been on how these pathogens can impact the host and how different metabolic processes then cause disease in this host. But we know that this isn't the only picture here, because pigs and ourselves are living in this highly pathogenic microbial world. And the question is, how come some people get sick and some do not? And really, what it simply boils down to is that we have all these defense factors in our bodies, immune, microbial, and these protect us from these pathogens and diseases. However, what's critical in determining whether or not animals get sick is this environmental influence. And we know this. We know that factors in our environment can alter the host and make them more susceptible to diseases. And one of the biggest factors that we study is stress and how stress changes the host. So if you look in animal production, our pigs particularly undergo a number of stressors in animal production, and these are inherent production stressors, and they can include weaning, dietary changes, and all these different factors that the pig is subjected to. And as a result, we know that many of these stressors can trigger GI disease. And when I talk about GI disease, this can be from range from suboptimal feed efficiency, so impaired nutrient utilization, all the way into maybe an infectious disease outbreak. But although we've known that stressors have affected our gut for probably 100 years, the mechanisms of this biology and interaction between the stress and the gut still remains poorly understood. And this is the big question. How does stress influence GI disease and GI disorders? And this is the focus of my research laboratory now at Michigan State University, where we really take a basic science approach to trying to understand the physiology of animals that are undergoing stress, and specifically how this impacts gut function. Because we believe that if we can learn more at the basic level, then we're going to potentially discover targets that we can bring to a very applied level in the animal. So what I will focus on this morning is we're covering stress and gut health, and I'm going to talk a little bit about stress physiology and the stress response. And then I'm going to focus mainly on early life stressors and how they can impact long-term gut barrier health, because a lot of the focus is on short-term effects of stress. My lab is looking at the impacts on the long-term development in gut function. And then I'll cover a little bit about what we've been learning about the mechanisms that are underlying gut function in the weaned pig. And then some summaries and take-home future directions. So the stress response. We talk about stress, and this is sort of a nebulous term, but when you look at the definition, it can apply to many factors. So this is one of the hundreds of definitions. A stress response is a reaction to a stimulus that disturbs physical or mental equilibrium. And this could be many things. We talk about the HPA axis, which I'll cover, but any type of stressor to the body, it could be a bacterial infection, an inflammatory response is a stress response. And what the goal, we talk about stress responses as mostly negative, but a stress response is critical. It's critical for survival. It's this very primitive system that has remained for as long as we've been alive and animals have been alive, that its goal is for survival. And if you look at the most well-studied stress response system, this HPA axis, we know that there's a coordination between the brain and all these peripheral nervous systems through the hypothalamic pituitary adrenal axis. And what all this does, this is a homeostatic mechanism to bring the body to be able to respond and meet this challenge of this stressor and then return it back to normal. So stress response is a good thing, it's necessary. However, when we get into trouble is when these stressors become, or we have an inability to cope or respond to appropriately to these stressors, or whether these stressors can be overwhelming or traumatic, that you cannot, their stress response is not effective. Or more commonly in animal production and even in people, these chronic stressors, these persistent stressors in the body where your body is trying to respond for survival in the fight or flight response, but it's these low-grade persistent stressors that wear down the systems. And another important factor about stress is the type and timing of the stress really matters. We know that what happens in the acute stress response can be quite different for when the stress is chronic. And we also know now that the age at which this stress occurs can have a huge impact on the stress response. And we're going to be focusing on early life stressors because there is a direct link between early life stressors and early life adversity and long-term GI and a number of disorders, not even GI. But in pig production, it's during the early life is when most of the most profound stressors are put on these animals. So that's why I'm going to focus on that. But we also know that these other factors in stages of life, there's different factors with immune system, microbiome, or microbial changes that can impact how the animal or person responds to stress. We have been focused on this weaning period in production as an early life stress because it is probably the most profound stressor an animal undergoes in production. There's these piglets, they're with their sow, their moms, they're nice with their littermates, and at a very early age, they're put in this environment, a weaned environment. And in this environment, there's all these compounding stressors. And even though these environments are as clean as possible and we take very good care of these animals, there are just inherent stressors that we cannot control. And they're quite dramatic if you look at all these types of stressors that are being imposed on this animal. And as a result, around this time of weaning, this is a period where there's a lot of disease. We know there's a lot of gut disease, and there's also a lot of respiratory diseases. So if you look at the gut and how it develops, we know that, and this is pretty similar to most species, we know there's a tremendous amount of prenatal development in the gut, but then this postnatal period, the first 1 to 2 months of life, represents a huge traumatic period of gut development here. And these include development of the immune system, establishment of the microbiota, development of barrier function, development of the enteric nervous system, and epithelial transport function, so nutrient uptake. In nature, when weaning pigs, it occurs gradually. It occurs 12 to 14 weeks of age. It's a gradual process. And you can see that it occurs during when the gut development has already predominantly been established. But if you look at what current weaning practices, occurs in this range. And this is critical because you can see these weaning practices are occurring in these critical stages of postnatal development. And again, what is happening here, this developmental phase, is going to shape the long-term function of this system. So if we look at what do we know about gut function at weaning, and there's been a tremendous amount of work here done about weaning, and I've just sort of generalized these changes. We know that upon weaning, there's acute inflammatory responses. We've shown some mast cells that are activated, and I'll go into what they are doing. There's changes in morphology, changes in digestive enzymes, microbiomes, different types of stressors. And what these do is they lead to these alterations in the normal gut function. And they could be barrier properties, secretory activity, changes in absorptive function. And the clinical outcomes are impaired performance and increased disease susceptibility. So I will focus on this important function of the gut, which is this barrier function. And this is critical. It's critical to animals, it's critical to any species. If you look at this gut, this is a histology micrograph of the intestine. You see these single layer of epithelial cells that line the gut here. And this epithelium is critical in that it must serve as an absorptive organ to transport electrolytes, absorb nutrients, digest nutrients. But at the same time, it must serve as a selective barrier to prevent the trillions of toxins and bacteria that reside in this lumen from getting across the gut. And this is particularly important because literally micrometers away from all these microorganisms is the largest immune organ in the body. So you can understand how important the integrity of this barrier is. So if you look at some of the aspects of this intestinal barrier, there's many components of it. There's extrinsic factors, which include secretory products, specialized cells that secrete mucus, and antimicrobial peptides. As Mark indicated, obviously a big emphasis on the microbiome and microbes and what are they doing, interesting aspects of how they're playing a role in barrier and host immunology and physiology. There's tight junction proteins, and these are important because these are proteins that regulate how permeable or leaky the gut is. And of course, there's an immune system that must be here, innate immune system that must rapidly respond to any type of breach or stress to try to return the body to homeostasis. And the enteric nervous system, which Mark also emphasized, and this is critical here because this enteric nervous system controls many aspects of all normal gut function. So what happens when this barrier becomes breached, which occurs during a lot of stress? And so you get leaky gut, and now you have all these antigens, pathogens, and toxins now able to freely traverse this epithelium and trigger inflammatory responses. And inflammatory responses in the gut can have a number of different effects. It can impair nutrient transport mechanisms, which I'll go into. It can allow infections to develop. It can activate neurons and cause a number of different pathologies. And in the worst case scenario, if it's severe enough, if it gets these bacteria and toxins get into the systemic circulation, then you get into multiple organ dysfunction. But if these stressors or barrier function become chronic, then you often see these stages of immunosuppression, where there's a chronic activation of the immune system. The mechanisms are still poorly understood on how this happens, but then this is another layer that may predispose you to infections. And just briefly here, the barrier function, how it relates to nutrition and nutrient absorption here. We know that a lot of these factors are transporters that drive nutrients across the gut, including energy, glucose, and amino acids, require a gradient to drive the nutrients across the gut. But when the gut becomes leaky, it loses some of this driving force. So you start getting some impaired nutrient digestibility or absorption. And we know that the barrier is so critical to many disease states. So impaired barrier function, just a number of all these diseases have been linked with a leaky gut. So it's really critical function of the gut. And that's why we have been really focusing on this in understanding how stress impacts this barrier. In our lab, we utilize an Using chamber technique to measure intestinal permeability. There are many ways to measure intestinal permeability, but we use this technique. It's the most sensitive, where we can take tissue from an animal that has been stressed, maybe weaned, and put their intestinal mucosa in this little slider system, and then put it on this chamber system. And if you look at this chamber system, the tissues in here, you have a luminal side and a serosal side. Tissue is oxygenated, it's warmed, there's nutrients in here. So essentially, we're just supplying what it's missing, which is the blood supply. So the gut in here can remain alive for up to 7 hours. And in the system, it can transport nutrients, it can respond to bacterial pathogens, it can modulate its barrier properties. So it's just an ex vivo system that very sensitively monitors the changes. And how we monitor the changes is, one, through these electrodes, which can measure electrical properties, and that can measure different transporters. But we can also measure the leakiness of the gut by applying probes that get through the paracellular space or the tight junctions, these labeled probes. We measure how fast they get across, or how much they get across a gut. And just for the sake of my presentations, I'm going to present a lot of this permeability flux data. So a high flux rate means a leaky gut or impaired barrier function, and a low flux rate means an intact barrier function, to help you set up when I start presenting my data slides. So one of the first questions we asked was about this weaning stress. This is such a stressful period in the animal. What is the impact on gut permeability? So we looked at intestinal permeability in weaned, unweaned pigs in their age-matched litter-mate controls 24 hours post-weaning, put their intestine on the gut, and show that just the event of weaning has this significant influence, increased gut permeability from this weaning process. And we've gone on to study some of the mechanisms that are driving this, and what we find is that there's immune cells that are activated and some stress activating systems, but feed intake during this time really doesn't have a predominant role during these early periods, whether they ate, whether they didn't, on this barrier function. I'm not saying feed intake does affect barrier function, but in these studies, this is mainly an acute neurochemical, neuromediated stress response. So then we asked, well, since this is such a critical period in development, what is the impact of weaning age? Does this matter? So we asked what if we move the weaning age up a little bit more, where the gut is a little more developed, does that impact how the gut responds to this weaning stress? So if you look at the permeability, again, this is an early weaned pig, an 18-day pig. Again, 24 hours after weaning, you have this increase in intestinal permeability. But if you look at a pig that was weaned at 28 days of age, just a little bit compared to their age-matched litter-mate controls, this permeability defect is markedly diminished. There's still some permeability issues with even a late weaned pig, but you can see the dramatic reduction in the gut. And we then asked, well, maybe it's just that later weaned pigs are less stressed. They can handle this stress better. So one way that we can measure, it's not the most sensitive measure, but it's well accepted, is this cortisol response. Because we know that any stress in an animal, they're going to respond with this HPA axis response and have elevated cortisol. That's why it's such a universal marker of stress. And as expected, 24 hours of weaning, these weaned pigs do have elevated cortisol. They're stressed. What about the late weaned pigs? They have elevated cortisol too. And it's actually even higher than these early weaned pigs. But the take-home point of this was that even the late weaned pigs, they perceive this weaning stress as quite stressful, and they respond with a stress response. So it's very similar, but the influence or impact on their gut was markedly different. So then we asked, given these changes that are occurring during this critical developmental period, what are the long-term effects? Because a lot of the work we study is mainly what's this post-weaning period. We kind of do the first couple weeks of post-weaning until growth rate picks up, feed intake picks up, some inflammation resolves. But we never really look at the long-term impact of this gut. And that's what we've been focusing on, because we want to know, maybe, these early life stressors, how do they maybe potentially impair the overall gut development, or maybe these animals do catch up eventually. So one of the first studies that we did was looking at intestinal permeability over time into adulthood. And we took different time points, post-weaning, pigs weaned at late weaned, 28 days of age, versus early weaned pigs, and we mapped out their permeability development. And what we found was these early weaned pigs, they had this robust permeability early at weaning. It resolves a little bit, but it remains persistently elevated throughout the lifetime of this animal. So just that early life stress leads to persistent increases in intestinal permeability. Then we asked what is the impact of the early life stress or current weaning practices on subsequent responses to this animal. So we did a couple of studies. One was a mixing stress study. This is a routine stress that animals undergo. We looked at late weaned animals, early weaned animals, grew them up, same environment, same nutrition, and then looked at how they responded to a mixing stress later, and then measured their barrier function. And what we found here, if you look at gut permeability, the pigs that were early weaned and exposed to this mixing stress had an increased intestinal permeability that was measured within 3 hours. So this is a very rapid process. Whereas the same pigs that were late weaned, subjected to the same stressors, did not exhibit this intestinal permeability. So it was affecting the long-term responsiveness of their gut. And this is fecal scores, blinded fecal scores as well, showing that there's also some functional differences that relate to some clinical differences as well. Another question we asked was whether or not early weaning or early life stressors can impact the subsequent immune or responses to an infectious pathogen later in life. And we did this in a post-weaning E. coli challenge model, where we took early weaned pigs and late weaned pigs. And in this study, these pigs, late weaned pigs, were about 23 days of age. That was considered a late wean for that trial, as compared to about a 16 to 70 day weaned pig. Grew them up to 35 days of age, challenged them with F18 E. coli, and measured the clinical response here. And what you can see is that the early weaned pigs responded to E. coli with a significant increase in diarrheal score. Late weaned pigs challenged with E. coli, they did develop diarrhea, of course, but it was less severe and less in onset. And if you look at their gut function here, this is permeability 4 days after this weaning, after this challenge. The early weaned pigs had still this marked intestinal injury, whereas the late weaned pigs had recovered. If you look at the immune response, in these animals. This was interesting because we know that the immune response is necessary. We need a robust immune response to respond and clear pathogens. And so what we found was that the immune response here was markedly suppressed in early weaned pigs, cytokines and immune cells, suggesting that there's some impaired recruitment of immune cells. Another thing that we begin to look at now, looking about nutrition and feed efficiency, is transport mechanisms. And we do this by measuring nutrient glucose-mediated transport across the gut. And if you look at an early weaned pig 60 days post-weaning compared to a late weaned pig, there's a dramatic influence on how the gut is going to respond to nutrients as well. So there's all these major functional changes in the gut that are occurring. And this is also similarly what happens in chronic stressors in animals. When they're under chronic stress, they have an impaired nutrient absorptive function. So in summary, just some of these changes in gut function, we have immediate and long-lasting changes in barrier function. We showed the heightened later life responses to stressors, dysregulated GI mucosal development, heightened enteric nervous system activity, and also impaired transport mechanisms. So then I'm just going to briefly go over just some of the mechanisms here. So we know that there's these early life stressors in weaning can result in these impaired development of the barrier and all these. What are some of the mechanisms behind here? And we know it's not inflammatory because the early wean and late wean gut is predominantly pretty similar histologically, despite developmental or functional differences. Histology is similar, not a lot of inflammatory cytokines, except when provoked. But one thing that we did find in the gut is elevated intestinal mast cell numbers. And these mast cells aren't picked up on normal H&E stains, but if you look at the number and the quantity of mast cells that accumulate in an animal that has undergone an early life stress, such as early weaning, there's a bunch of mast cell activity. And we know that mast cells are allergic-mediated immune cells. They mediate a lot of allergic reactions. That's kind of what we know them as, the potent inflammatory functions. But we also know, becoming more known, that they're a central effector cell in the stress response. And they do this by releasing mediators into the surrounding tissue that can affect a number of functions in the gut. And this is just an overview. They can induce inflammation, change absorptive function. They can also interact closely with enteric neurons to drive changes. And this is interesting in irritable bowel syndrome in humans, a disease that's linked with early life adversity. They also have elevated mast cells. This is a pathologic hallmark in disease. So what is the role of mast cells in the pig, early wean pig gut? And to get at this question, we tested the effects of this mast cell stabilizer drug, sodium cromolyn, administered it to the pig, the early wean pig, to see if we could change intestinal permeability. In 24 hours after the treatment with cromolyn, we were able to dampen and restore normal intestinal permeability mechanisms. And what this means is that the mast cells are playing a critical role in modulating this stress response. And we've also shown this work earlier in the post-weaning period. And this is some mouse data, and I'm only showing this just as another proof of the role of mast cells. In our lab, we can look at mice that are mast cell deficient, and we can subject them to stress and look at permeability. This is a control animal, wild type. They have increased intestinal permeability, but mice without mast cells do not exhibit intestinal permeability. And then when you give mast cells back to these animals, you can restore it. So what this says is that mast cells are these critical stress effector cells in the gut that we're really beginning to look at now. We've established some of the mechanisms by which mast cells regulate permeability, and they do this through the release of these mediators that can influence intestinal permeability. And now we're trying to understand how they're regulated in stress responses. So what we're asking here is that we know this, in summary, this natural weaning process is of this normal gut development. And I call this now gut health potential. This is the natural situation. But now as we're looking more about our current weaning practices and early weaning, we're seeing that there's some marked changes in how the gut develops. And if you late weaned a little bit, you can improve this a little bit. But what I'm looking at this as a lost potential. You know, these are lost immune potential that potentially we can get back. And that's what we're trying to understand, some of the mechanisms here that will influence this trajectory of gut development, early life interventions. And we know that in production, weaning age is determined by many factors. We can't just wean pigs at 12 weeks of age. Some production sites can't wean more than 25. It's not economically viable. But the point here is that we're looking at a potential loss of gut immune and gut health potential here, that maybe we can still wean early, have some interventions, and then drive development of this gut. So obviously, I'd like to thank the advisory committee for inviting me to talk here. And this work is done by a number of former and current lab members that were critical for this work to be done, and our collaborators. And with that, I'd like to take any questions you may have.
Thank you, Ed.